Everybody Namaskara. Today we'll be dealing on the topic Anthoceros reproduction, both vegetative, basic, sexual, and the sporophyte. I'm Dr. Jayalakshmi, Assistant Professor of Botany, Postgraduate Department of Botany, Marine Science College, Pullman, Mysore. So this is a topic in first and BSc of the syllabus of University of Mysore. Coming to the objectives which is dealt in this presentation is the general features, that is reproduction, vegetative, sexual, and the sporophyte and the alternation of generation found in Anthoceros. Coming to vegetative reproduction, it takes place by the following methods. That is the progressive death and decay of the thallus, by tubers, by production of gemma, and by persistent growing apexes. Death and decay of the thallus is not very common method, more common in liverworts, where a part of the thallus decays and the remaining part, which the lobes which separate out, will develop into a new gametophyte. By tubers, so this is very commonly found in Anthoceros. So while some of the species like Anthoceros himalayensis were very well established in producing these tubers. So tubers have outer protective corky layer and resist extreme desiccation. They remain alive even after the death of the thallus. In sufficient moisture, they germinate and give rise to a new plant. So these tubers usually will keep the thallus alive during the propagating structures during the unfavorable conditions, and during the return of favorable condition, these tubers germinate with moisture, giving rise to a new plant. Another type of reproduction, that is the production of gemma. So some species like Anthoceros glandulosus, Anthoceros formicae, and Anthoceros fusiformis that bear some stalk structures on the dorsal surface called as gemmae. These are produced in a cup-like structure called as gemma cup, which has a fringe margin producing several gemma, and each single gemma is in somewhat oval-shaped structure with a notch in the sides and on separating out from the gemma cup and falling on a suitable substratum, they give rise to new plants. Another method of vegetative propagation by persistent growing aphids. Species like Anthoceros personae and fusiformis will reproduce by this persistent growing apexes, mostly in areas where summer season is very dry. All parts of the thallus except the apical region becomes dry. These apexes later, after a period of dormancy and with the return of favorable condition and sufficient moisture, these apex which was having some few life cells will give rise to a new thallus. So these are called as persistent growing apexes. So these are the different methods of vegetative reproduction found in Anthoceros. Coming to sexual reproduction, sexual reproduction is always oogamous. The male gamete is a small and a motile one, while female gamete is large and non-motile. The male reproductive organs are antheridia and the female reproductive organ is archegonia. Both male and female reproductive organs are multicellular and they are provided with a jacket. Antheridia are differentiated into stalk and body, while archegonia are stalk and differentiated into venter and leg. So coming to a detailed structure of antheridia, the antheridia occur endogenously. So the antheridia here is embedded within the thallus, that's why it's called as endogenous origin, especially on the dorsal surface in a closed cavity which we call it as an antheridial chamber. They enclose mass of androcytes which mature, that is the androgonial mother cells will give rise to androcytes which will metamorphosize into biflagellated antherozoa. So this is the antheridial chamber which produces one to a few antheridia within the same chamber, and each of this antheridia is provided with a multicellular stalk and oval-shaped body which is jacketed. Within that we find the androcyte mother cells or androgonial cells which will divide and give rise to a sperm or an antherozoid, which is a highly coiled structure provided with flagella. The cilia are attached to the anterior end of the body. The antherozoids usually are released by the bursting of the antheridial jacket wall, and they swim in the water by the lashing movement of their flagella and reach the archegonia. Coming to the female reproductive organ, that is archegonia. A mature archegonia is a flask-like structure without neck canal cells and with an egg. So you find this is the mature archegonia is a flask shaped structure. So you find a ventral portion here, which is a bulge, and an elongated structure, which is called as a neck, surrounded with neck canal cells. Provided at the tip of the neck is the cover cells. At the top of the neck of the archegonia, there are usually four cover cells, which become separated when the archegonia mature, and by the gelatinization or the disintegration of the ventral canal cell and neck canal cell, the mucilage produced appears at the neck of the collar, the cover cells, and which this mucilaginous content attracts the sperm cells, and hence the sperm cells are get chemotactically attracted and enter into the neck, reaching the egg for the process of fertilization. The fertilization occurs when the sperm cell comes and fuses with the egg, leading to the formation of a diploid zygote. Now, the diploid zygote gives rise to a sporophyte, the next generation of the Anthoceros. The sporophyte of the Anthoceros has this following structure. That is, it's usually born on the gametophyte. So, the archegonia, which is embedded within the thallus of Anthoceros, will now give a place for the development of this sporophyte, that is, the zygote or the product of fertilization gives rise to an embryo which develops into a sporophyte. A mature sporophyte, in the case of Anthoceros, is not totally dependent on the gametophyte because it's partially independent, as it has the cells have chloroplast and can photosynthesize. A mature sporophyte, if we see, consists of bulbous foot, a smooth, slender, erect, cylindrical structure called capsule, and the capsule varies from 2 to 15 centimeter in length in different species. So the sporophyte is a structure where we find a foot which is embedded within the thallus. Surrounding the basal part of the foot, we find a fringed margin of the thallus, that is what we call it as involucre, which protects the basal part of the sporophyte, and that region of the sporophyte consists of the meristematic zone. Followed by that, we find erect, stiff, horn-like structure, which is called as the capsule. So, coming now to each of these parts one by one. So, the sporophyte has foot, seta, and capsule. These sporophytes appear like bristles or horns on from the surface of the thallus. Hence, the Anthoceros are called as hornworms. Coming to internal structure of all these parts, the mature sporophyte is differentiated as I told into foot, seta, and capsule. The foot is a bulbous multicellular structure which is made up of mass of parenchymatous cells, and it acts as an ostoria which absorbs the food and water nutrients from the thallus or the gametophyte and provides it to the developing sporophyte. Followed by that is the meristematic zone or the intermediate zone, or we usually call it as intercalary zone. Seta is represented by meristematic zone in case of Anthoceros. So seta is meristematic zone or intermediate zone is replaces seta, while which is found in other bryophytes. So this is seta is represented by meristematic zone. This is present at the base of the capsule and consists of meristematic cells. These cells constantly add new cells to the capsule at its base. So the sporophyte of market of this Anthoceros grows continuously until the food gets exhausted. The presence of meristem at the base enables the capsule to grow for a long period and form the spores. This is a unique feature of Anthoceros, which is not found in any of the bryophytes, and we are able to see different stages of development of the spores from base upwards in the sporangium of Anthoceros. So we find a meristematic zone, then the formation of archisporium, then we can see the meristematic zone, then the formation of archisporium, Then the archisporium forms into spore mother cells. That spore mother cell gives rise to spore tetrads, and that spore separates out into spores and pseudo-elaters, and then they get liberated. So in a single uh, mature sporophyte, we can see all these stages because that the meristematic zone has a property of division and continuously divides and produces new cells, and old cells get metamorphosized and matured as it moves upwards. So capsule is the major part of the sporophyte, which is differentiated into columella. So columella is a central sterile running portion of the sporophyte, extends nearly to from the base to the tip. It is endothelial in origin, and in the young sporophyte, it consists of 4 vertical rows of cells, but in a mature sporophyte, it's made up of 16 rows of vertical cells. In a young one, that is, we find usually 4 cells. That 4 cells will divide to form 16, a group of a square of 16 cells which run from the base to the tip of the sporophyte. So in a transverse section of these cells, that's why it appears as a solid square and provides mechanical support, functions as water conducting tissue, and also helps in dispersal of spores. Since the columella runs from the base to the tip, it provides mechanical support and supplies water and nutrients to the upper surface of the sporophyte. And also, when the sporophyte is mature, the columella helps in dispersal of spores. Next is the region followed by the meristematic zone. In the capsule is the presence of archisporium. So it extends from the base to the top of the capsule. So a few species of Anthoceros here, the archisporium may remain one cell in thickness throughout the further development. So you find the archisporium which is found after the meristematic zone. The meristematic zone gives rise to archisporial cells. It may be one layer or it may be 2 layers in thickness. In some species like Anthoceros personae and Himalayensis, it becomes 2 layered thick and a little above the base. And in other species, even it may become 2 to 4 cells in thickness. While above that, we find this archisporial cells gives rise to the spore mother cells. So this archisporium cells will give rise to the spore mother cells. The spore mother cells will undergo reduction division, giving rise to spore tetrads, while some of the spore mother cells give rise to a single filamentous structure called as pseudo-elaters. So pseudo-elaters may be unicellular or multicellular, branched or unbranched structures which consist more or less elongated cells. The spiral thickness is absent in the elaters of Anthoceros, and in some species you may find secondary wall thickness in this. Hence, they are called as pseudo-elaters, since they lack the spiral thickness or hygroscopic movements. Coming to the upper portion of the sporophyte, so we find spore tetrads which will the spore mother cell which undergoes reduction division and forms haploid spores. So haploid spores fills the cavity, uppermost cavity of the capsule, and once the capsule is mature completely, then the capsule will rupture at the apex. So at maturity, the tip of the capsule wall loses water. The loss of water from the capsule wall will be very helpful in the digestion of the capsule. After drying of the capsule, the slit appears just below the tip, which gradually widens downwards, and hence the capsule wall will split into 4 halves. At the apex, the hygroscopic movement of the moisture and the pseudo-elaters which are present there in the upper portion of the capsule, and air currents at this stage of this, all of these will help in the dispersal of the spores. The tip of the columella projects out like a flagellum, and under favorable condition, the spores are immediately released from the capsule, and they fall on suitable substratum and germinate immediately, or sometimes they may go under period of dormancy or rest, and then they germinate, giving rise to new gametophyte thallus. So hence This spores which are released from the sporophyte will undergo the spores which are haploid will germinate to produce a young protonema which gives rise to a new gametophyte. So the life cycle of Anthoceros shows regular alternation of two morphologically distinct phases, that is the haploid phase or the gametophytic phase, which is the dominant phase and produces sex organs, and sex organs produce gametes leading to formation of diploid zygote after fertilization. That fertilized zygote will give rise to the next stage, that is the diploid phase or the sporophytic phase. The zygote develops into a sporophyte, and the sporophyte is represented by an elongated horn-like structure. So these two stages alternate consecutively one after the other, which complete the life cycle of Anthoceros. That is why we find the alternation of gametophytic and sporophytic generation in Anthoceros. So these are the books and sources of references to prepare this presentation. Thank you.